Okay, welcome everyone to our final seminar in the SNARL Spring Seminar Series. And Adam gets to the distinct honor of bringing in the final talk of the spring. And I just wanted to say too that Adam is one of our very own graduate students. He's worked here at SNARL for many years. He was a grad student here when I first started at SNARL. And Adam is also a recipient of our Valentine Reserve Fund Graduate Student Fellowship. So Adam has done a lot of work here, and he has actually benefited from the generosity of our donors. Several of you are probably on this call. So it's really nice to have Adam come back and speak with us. And Adam is not only graduated and moved on. He's graduated and moved on, and he's returned to the Eastern Sierra to do his field work. So he's actually here in the Eastern Sierra right now doing his field work. So it's my pleasure to introduce Adam Cohen. Adam has been drawn to Sierra Lakes since his first backpacking trip into the range at age 10. This directly led him to his doctoral research at UC Santa Barbara, focusing on the impacts of high Sierra reservoirs on water chemistry and benthic macro invertebrate communities. His graduate fieldwork was based out of SNARL and would not have been possible without a field station. Adam has since joined Stillwater Sciences, where he continues studying Sierra reservoirs in the context of hydropower relicensing. And I know I and many of you guys out there are interested in hearing more about hydropower and what's going on in that the world, especially in the Eastern Sierra. So Adam, thanks for joining us, and I'll turn it over to you.
Sure. Thanks, Carol. Yeah, I was going to bring this up at the end, but it's worth saying again that I very much benefited from being able to go to SNARL, and absolutely would not have been possible to do the field work that I did without having SNARL as a field station and the lab facilities. And I hope to also be able to pay that forward someday in my future too, because it's just so important to being able to do work in the Eastern Sierra. So like Carol said, I'm actually, I'm back again. I'm in June Lake right now. We're doing field work up in Lee Vining. So I guess a disclaimer or just FYI, I'm with Stillwater Sciences. We're working with SCE now on the Lee Vining relicensing, and to a lesser extent, though I'm not part of it, on the Bishop Creek relicensing. We're not involved with Lundy or Rush Creek, but I'll talk about that more later as we get into that. But I'm currently just got back from sampling up at Tioga and up and down Lee Vining Creek earlier this morning. It was it's pretty cold up there, and Saddlebag is still frozen. Kind of amazing for almost being June. Let me go ahead and share. So I am going to give you a couple things here. So I first want to give you a little intro into relicensing, and then I will step back and talk about my dissertation work that I did at SNARL, which is very much directly relevant to the relicensing going on now. And mostly I'm going to focus on the Lee Vining project, but we can try to pick my brain on the other ones as well. But I know the most about Lee Vining, and it's where I was today and go back to the most. So I'm going to talk a little bit about relicensing, go back to my dissertation work, and then come back to relicensing at the end and see where in the process each of the Eastern Sierra relicensings are. And I've tried to keep this high level. If for some reason I get too into the weeds on something, please stop me. If I use words you don't, I've tried not to use any very jargony words. That's take some time to get that out of your system and make everything understandable. But if I do, please stop me and I will back up and explain anything. And I will do my best to try to follow questions as they come. Though, Carol, I'm just realizing that with the screen shared, I can't see questions. So if somebody has something that I'm not responding to, Carol, you can just bug me and stop me. So a little background on Sierra Nevada lakes. There are thousands of them, obviously. If you live around here, you've probably seen them, you know that. Part of what's maybe special or important about them is that they're very new, geologically speaking. 12,000 years, believe it or not, is very, very new. They're very young lakes. So part of what that means is that there's very little going on in the watersheds. They're very granitic, like you can see in the background here. This is Spooler in the background. And I probably will try to point out each of the lakes as I go here because I'm just excited about pretty much all of them. So this spooler in the background here just give you a sense of what most of these look like. They're mostly small, they're relatively shallow. So, you know, may feel deep to us, but they're relatively shallow as far as lakes around the world go. There's no native fish, despite what it may seem like, except all the way down in the Kern. And with Stillwater, we've kind of started to dip our toe into that a little bit, but at least here in the Easterns, there's no native fish. And in terms of native aquatic vertebrates, we're limited to some snakes and frogs, and some of the SNARL folks might correct me, but or native aquatic vertebrates, we're limited to a handful of species, and the frogs are being reintroduced, so we're starting to see them in more places now. But all the fish you see were pretty much all planted. These lakes are ice covered most of the year, as evidenced by Saddlebag still being frozen today. And even in years like this, where it's really dry, most of the precipitation occurs as snow, especially it seems like up at high elevations in years like this. Even if below, say, 9,000 feet didn't get that much snow, there's a good amount of snow above 10,000. So just as like maybe a sense of what these watersheds look like, this is, if you're familiar with the area, this is Little Lakes Valley looking down at, you can see Long Lake on the right there, and then the valley extending onto the right. Just a very general idea of what these watersheds look like and what we're talking about. And in general, the watersheds that the reservoirs are in the Eastern Sierra are really not that different from these lakes. So it provides a good natural experiment to set up a comparison between natural lakes and reservoirs. So a little bit of very basic information that is really useful to know for the remainder of this talk. In the Eastern Sierra, this can be different elsewhere, but in the Eastern Sierra, natural lakes and reservoirs differ in this very important way, and this will be relevant throughout here. Natural lakes, obviously, are just releasing water off of the top of the reservoir, or off of the top of the lake. It's just excess flow, essentially, that comes off of the top. Whereas the reservoirs are managed and dammed, and the water is being released from the bottom. Obviously, in some cases, the reservoirs spill, but that happens less and less frequently. And even when they do spill, like say Saddlebag or South, that they do spill, just not that often. It's pretty minimal compared to the release coming out the bottom. So not only is the flow from the reservoir managed, it's important that it's coming from the bottom of the reservoir versus the top of a lake. And this next slide give you a little information as to why that matters. So this is just a basic diagram of what happens in basically every high Sierra lake, is that there's layers through the summer. So as the top heats up, it's separated by density, essentially, from the bottom, which will separate them. Any chemistry may start to differentiate between the top and the bottom in both the summer and the winter. So if you were to take a sample from the top versus the bottom in the summer or the winter, it may start to actually look different. Versus in the fall, there's going to be short periods. And in the spring, there's going to be periods where the lake is the same temperature, top to bottom. So it turns over, which is what those turnover figures are. In spring, that happens after ice off. In fall, it happens when the lake cools down enough that it's 4 degrees top to bottom, because water is actually most dense at 4 degrees. That's why you see in winter, the coldest water is actually at the top. So the reason that prior figure was important is that in the summer and in the winter, that bottom water that can become sort of physically separated is released from reservoirs, whereas it's not from lakes. So obviously I said that there's thousands of lakes in the Sierra. There's also thousands of reservoirs in California. Many of these are small, but some are really large. Ours in the Sierra and the Eastern Sierra in particular are not actually that large as far as reservoirs go, but they're all pretty new. So for the most part, they're about 100 years old and provide a variety of services. Some are, mandated by the Forest Service to provide some recreation now. So they're not their original intended purpose, but they've become multi-use and provide more benefits that they were originally constructed for. But they also act as barriers to a variety of things, including fish passage, though that's less of a concern in this part of the Sierra because we don't have any native fish. Focusing on Eastern Sierra reservoirs for a second, Saddlebag on the top and South on the bottom. They're all about 100 years old. They're mostly built in the 20s, give or take, say plus or minus 5 years for the most part. And they're almost all were built to supply power originally to nearby mining towns. And then as the generation capacity increased and the mining towns sort of started to die down, they started to provide power to Southern California. Today, they still provide power to California, and they now provide access to recreation, water supply for various uses, and they're also managed to provide very consistent in-stream flows in the streams they're damming. So I think this picture on the top right is a Bennett Bill, if you've ever done that hike up out of Junction Campground. It was the little mining town that was at just above Tioga Lake. And that was basically the impetus for the construction of the road that's up there, and then at least partially related to the construction of the Levining project, which is what I mostly am going to focus on. And you can see on this map from 1901 here, Ellery, Tioga, and Saddlebag are all natural lakes, and that's actually pretty typical for Eastern Sierra reservoirs, whereas a lot of the West Slope reservoirs are basically just big rivers that were dammed. Eastern Slope reservoirs are frequently existing lakes that were dammed to enlarge them and increase the capacity for hydropower. So you can see on this map before the dam was, any of the project was actually built, you can still see those same lakes existed. So jumping into the relicensing process, this is all managed by FERC, which unless you're involved in relicensing, you've probably never heard of FERC because it doesn't really touch on your daily life unless you are in one of these worlds. It's a kind of a unique agency in the federal government. And at least in my capacity, I don't deal with it in anything other than relicensing. But it is the agency that entirely manages relicensing. So every large reservoir needs to have a license from FERC. And to get a license, there's a variety of requirements, which I'll talk about in a second. But this is just to say that that's all done, managed by FERC, though they make very few decisions on their own. They're meant to be kind of a mediator between the licensees. So in this case, it's mostly Southern California Edison and the Eastern Sierra, and then the assorted stakeholders, which mostly include general public, interested public, but mostly agencies actually. So Forest Service and say CDFW. And also these are very long processes. I guess maybe not that long in the scheme of government projects, but it takes maybe 5 or 6 years to get a new license, or I should say, relicense an existing project. So all of these projects in the Eastern Sierra have licenses that are just coming up for renewal. So this might be a little dry, but I feel like this is very relevant if you feel like getting involved in this process, because there's a lot of jargon. And if you start following along with the documents, this is useful to know. So the first thing that any of these projects will do will say, present all the existing information that's available for a project. That's actually a really useful place for stakeholders to, like the public or agencies, to get involved, because often SCE or myself are not aware of all of the existing information. If it's floating around in somebody's computer at SNARL, or I don't know, it's older data that's on paper somewhere that we just don't know about it, there's no way to know unless people tell us. So any existing information that's out there goes in this first document. And the idea there is to try to lay out potential data gaps. So what don't we know about this project? Not as just a blanket statement like general academic questions, but what's directly relevant to the management of the project that we don't know. And that leads into steps 2 and 3, which are, okay, if there are unknowns, what studies can we conduct here to try to address those data gaps within a very condensed time frame, which is you basically have 1 to 2 field seasons to do that. And the goal of doing that would be to try to provide information to set up license conditions down the line. So if you do see, say, an issue about something about the way it's managed in terms of geology or recreation or water quality or aquatic resources like fish, whether or not they're native, that's what those studies would be to inform. And then once those are conducted, you'll actually, or the SCE, the licensee, will submit an actual license application to FERC. And anywhere along the way here, people can submit comments. I don't think FERC at any point prevents you from submitting comments. And I'll come back to this at the end too, but substantive comments are always useful. And FERC's goal here is, again, just to kind of act as a mediator, and they make few decisions unless there is real disagreement, which can happen. But the idea is for them to just get the agencies and the licensee to talk to each other and work it out, essentially. So now I'm going to jump back to my dissertation work, which is basically feeds into step 1 of this process, which I didn't really know at the time. I kind of was hoping that it would be useful and somebody would find it helpful in the relicensing process, because I was aware of it as an undergrad and then starting grad school. And I kind of took these questions and that maybe they would be interesting and useful for that process. And turns out that they are very useful. And now, at least these first 2 questions, my work was incorporated into the process and has become part of the record and part of the relicensing process now. And so I'll talk about what those questions were and what we found. So this first question is very general, maybe kind of basic question about water chemistry in the Sierra. So this is thinking again about that figure showing lake releases and reservoir releases. How do lake and reservoir nutrient concentrations, so just focusing on nutrient concentrations for a second, rather than maybe some other components of water chemistry, for the reason I'll get into with question number 2, how do the nutrients concentrations differ, if they do, and if so, what's controlling them? So part of the reason this matters is if there's a difference in primary productivity, that's probably driven by these concentrations of nutrients. But we want to know if they do differ, why? And if so, is that related to that figure I showed for water being released from depth? So to address this, based out of SNARL for basically 3 years, well, 3 years on and off, wasn't there the whole time, but some great summers at SNARL. At each of the 5 lakes and 5 reservoirs in the Sierra, the reservoirs were Ellery, Saddlebag, Tioga, South, and Sabrina. And then the lakes were some natural lakes scattered nearby to each of those. And in each of those, we took profiles of temperature and dissolved oxygen. And that's, I'll show an example of that in the next slide, but that's basically just from top to bottom with an instrument like what I have here on Sabrina, is dropping the instrument into the water and then looking at a profile through the water column of each of those constituents. And then also, if you remember that figure with the layers, I was taking samples of water chemistry here just focusing on the nutrients and then chlorophyll related to productivity from the surface, and then the bottom, and then the outlet too. And I also, at each of the reservoirs is, SCE is recording discharge, so flow on their own. And for the lakes, since that data wasn't available, I collected the data myself, essentially. So installed some instruments and went and measured flow enough times that we could predict the amount of flow from just depth, so we could record it continuously. To give you an idea of the type of years, if you don't remember this, the type of years that I was sampling for, 2016 was my second summer and 2017 was my third. So I got lucky in terms of my study in that the water year types spanned a large range while I was out here. So this helped inform my results as I got a large range of flows and snowpacks. So if you remember, 2017 was a wild snow year. It was just a crazy amount of snow, which is a major, major contrast to 2016, where stuff was, basically dry by July. As you can see in this photo, the blue is the snow here. And in July, we went up to, I think, middle of July, we were still going up to lakes that we walked out onto because they were frozen solid. So I was lucky enough to span these, to be at SNARL spanning very, very different years. So this is an example of a profile in one of my natural lakes that I studied. The orange line is from 2017, the cold year, the wet year, and the blue line is from 2016, the dry year, from just 2 days apart, 1 year and 2 days apart. So this is, you can imagine, kind of like a cross section into the lake of temperature, as the warmest temperatures at the top, and then a little small thermocline, so a small decrease in temperature down to the lowest temperatures at the bottom. So you can see in 2016, on basically the same day in the summer, it's much, much warmer, and the lake was stratified, meaning that top water is starting to physically separate from the bottom water, whereas in 2017, the lake ice had just come off. So what does that actually mean, other than, sure, it was a wet year? So that means that when it's a wet year, warming doesn't start until much later. Stratification doesn't start up until much later, meaning the top and bottom layers don't start to separate until later. But that also means in a wet year that stratification also sets up under ice. So if you have a wet year where ice off doesn't happen until later, you have a longer period where the lake stays stratified or separated in the winter, which will have sort of similar effects to what happens in the summer. So just basically jumping into the results here. The controls on, and I'll kind of go through these quickly, but the controls on the nutrients were basically related to how long it's been since ice off, characteristics of the watershed, which makes sense. It's the inputs to the system, how long winter was, and then water residence time, which is kind of basically, the idea is how long was water sitting in the lake, which is strongly related to flows. I mean, it also is related to the volume of the lake, but it's basically pointing to the amount of snow that year. And these other nutrients that I looked at were pretty similar. It's the same idea, is it's related to the inputs from the system, so what the watershed looks like, and some characteristics related to snowpack, essentially. So what does that actually look like in terms of differences between lakes and reservoirs? So each of these three, this is just an example using nitrate, and it's just concentration on the Y. The units don't matter here. What matters is that although you see differences between lakes and reservoirs here, these are not significantly different. So statistically, they're not different in the top layer, the bottom layer, or the outlet, which is kind of interesting in that even though they are, they look different here, the spread is large enough that they don't actually come out as significantly different. But when you look at this plot all the way on the right, which is export, so that's saying, okay, there's this concentration coming out of the lake or coming out of the reservoir, but what if we also include the flow? So there's this concentration, but how much of it is being released? So it's concentration times flow. And when you put that together, the amount of this nutrient, in this case nitrate, that's being released from reservoirs is very significantly higher from reservoirs than from lakes. And this is getting back to what I talked about initially, is that the top and the bottom separates, and the reservoir releases water from the bottom. And so that those concentrations at the bottom, which you can kind of see in this middle plot here, hypolumion, those concentrations at the bottom are a little higher. And that water, particularly in the spring under ice, and then at the end of a long summer when stratification is set up, those concentrations at the bottom are higher. There's more nutrients at the bottom, and that's being released from the reservoirs and not being released from the lakes quite as much. And especially, and then when you factor in the amount of water being released, it becomes a really significant difference. So if you look at this across, so this is broken out by, there's a lot of information here, I realize, but this is broken out by year and then season, so you can see when this actually happens. And that's what I'm trying to get at here. The little asterisks show you where there's a significant difference. So you can see when the separation actually happens is, so like summer 2016 was really dry. So stratification happened earlier and extended for longer. So that difference between the top and the bottom set up earlier, and then you start to see a difference in the release in summer, and then it extended into fall. Whereas in 2017, it happened briefly in the summer and then disappeared by fall. So just to recap this section, lake and reservoir nutrients don't look that different. The inputs are really similar. They're controlled by essentially the same things. But the real difference is the release point and the amount of water that's being released, because reservoirs have license conditions. So they're required to release a specific amount of water, whereas reservoirs just do whatever's happening in the watershed. So the reservoirs end up releasing more of these nutrients, even if the concentrations are similar. So moving on to whether this matters at all for bugs or anything else living downstream, I focused on benthic macroinvertebrates because there's not a lot else that lives in the stream. So if you want to look at something that's native that lives in the stream below these lakes or reservoirs up in the High Sierra, you're limited to basically benthic macroinvertebrates. Though, like I noted here, frogs are starting to be reintroduced, but it's limited. And there's a lot of debate between people interested in fishing and people interested in frog restoration. So that's ongoing, but frogs are starting to be introduced. But the frog world is its own little world that I'm not really involved in. So I can't really speak to the mountain yellow-legged frog or the Sierra yellow-legged frog info. Inverts are also useful because they sort of aggregate information about water quality. Some groups of them are really sensitive to differences in temperature and water quality. So they're a good way to see whether there's a ecological response to differences caused by whatever's going on upstream. And they're also pretty easy to sample relatively, especially for a water quality person like me that doesn't know that much about vertebrates or invertebrates really. So I collected benthic macroinvertebrates in the same 3 summers, in the spring, summer, and fall, to try to capture differences across the season. We didn't want to just bias to a specific part of the year. Identified them mostly to genus, except for some groups, at SNARL. Spent many a long day in Dave Herp's lab at a microscope, identifying these bugs. And along with those data, we use the water chemistry that I talked about already, and then the discharge that I brought up and I will show you. So these are plots of all of my reservoirs that I was looking at, and all of the lakes where I was sampling. So the lakes are the data that I collected myself. The reservoir data is just from USGS, essentially. It's recorded by Edison, though. So you can see some major differences here are some of the red lines, the reservoir lines, are kind of on top of each other, so it's a little hard to see, but you'll notice that they stay well above 0 for a good chunk of the year, which is very different from the lakes, which go close to 0 or very near to 0 for a good part of the year. And then the other clear difference here is that 2015 was extremely dry, 2016 was dry, and then 2017 is just far and away much wetter. But other than that clear water year type difference here, the takeaway here is that the lakes follow really clear, typical Sierra hydrographs that are, you see snowmelt start, snowmelt peak, and then as the snow has basically melted, you drop down to 0. When the snow is gone, you're pretty close to 0 flow. There's still some water coming out of the watershed, but that's a very minor portion of the flow. So also just jumping into the results here, I think what I want to show here is that you see really big differences, one in densities across the years. So 2015 was really dry, didn't find a lot of bugs. 2016, a little wetter, found some more bugs. 2017, a lot more. And then you'll also notice that in 2016, especially 2017, this red box, which are the filter feeders, just take up, especially in the summer and the fall when flow really picked up. Those were just far and away the biggest component of the invertebrates that I found. And you could actually see this yourself if you're ever out on a hike, anything in a wet year. If you go to a stream and look at like a boulder where there's a good amount of flow coming over it and look at it, it looks like there's like little bits of dirt almost on the face of the boulder. But if you rub your finger on it or something, those are all little larvae. So it's worth, next time in spring you see a lot of flow somewhere, just look at some boulders and you'll see hundreds of fly larvae. And that's basically what we saw here, is overwhelmingly were fly larvae. So this is just one of the more technical parameters that we looked at for benthic macroinvertebrate diversity, or for benthic macroinvertebrate community composition. You can see it's often actually lower below lakes, which I don't think I have the plot in here, but that is probably related to reduced flow and the fact that many of the lake streams dry up for a portion of the year, whereas the reservoirs do not. I know there's a lot in this figure, and normally I hate to include figures like this, but there's some things that are important in here that I wanted to bring your attention to. One is that, so the way to look at this without getting into the stats details of a plot like this is that I've included the ones here that show you lake in red and then reservoir in blue. And so what you should notice here is that you don't see any clustering to one side or the other in either of the plots, is that the spread is kind of distributed evenly across both. It doesn't seem like or the statistics also agree that there's no clustering to one side or the other. And what that tells you is that the benthic macroinvertebrate communities are not different between the lakes and the reservoirs. So you also notice that across fall, spring, and summer, which are the different shapes, there's a little bit of a shift. You can you see them move a little bit, but it's also not significant. The other thing to notice here are the arrows on the left side. So D50 is actually the substrate size. So it's basically the size of the rocks in the stream where we were sampling. 1 max is the daily maximum flow. So basically the size of the snowpack that year. And then the other ones that are important there, or the most important one on below, is NH4F, which is ammonium flux, which is one of the parameters that I said was significantly different below the lakes and the reservoirs. So what matters here is that even though the lake and reservoir communities weren't different. They're still at least responding to some management of, or the effects of reservoir management. I'm gonna, for sake of time, I'm gonna skip through the recap here and go on to the last chapter, which was looking at CO2 emissions from lakes and reservoirs. And this was sort of brought up in the middle of my dissertation and realizing that this was a new and interesting idea that nobody was really looking at in the Sierra. There was basically no data. There's a little tiny bit of data on CO2 in Sierra Nevada lakes and none in reservoirs, and very little data on how much is released. So basically we don't know how much, I mean, you can make some guesses based on other data that are out there, but no information whatsoever on how much is being released, or really a better idea of the spread of what's out there. So this is a picture of a very frozen spooler with the instrument that I'll talk about in my boat. So we measured dissolved CO2 in all 10 of the lakes and reservoirs. And then this little chamber here measured CO2 flux, so the amount of CO2 coming out of the lake or the reservoir. So the amount, actually, you can imagine it, evaporating is not the right term, but if that's useful to think about, you can think about it that way. It's the amount degassing. Like if you say you opened a can of soda or beer or whatever, those bubbles coming up, exactly the same idea. And that chamber is meant to capture that and actually quantify how much is coming out. So that's just what this diagram is showing you. That chamber sits on the top of the water, seals on the water surface, and there's a little sensor at the top. And you do a little math to figure out how much CO2 is being released from the reservoir or the lake. So this is just a plot of all of the data together in all the lakes and all the reservoirs, starting from, so I've normalized this by days since ice off, because that varies so much across the gradient of my sites. So days since ice off could have, I mean, the actual date could vary from of 0, could be, you know, maybe May 1st at one, and then July 15th at another. So I thought the best way to think about this would be, okay, what is actually, when does this lake see spring? How does, when does spring actually happen here? And if you think about it as ice off, you start to see a really nice pattern here. If you just did this by date, you'd see no pattern whatsoever. It'd be very confusing. But if you look at the points before 0, those are samples that we took from under ice. So all of those, you notice, are above 100%. So let's imagine like the soda can or the beer can opening again. Those are super saturated. So those are the bubbles coming out of the lake or the reservoir. Not actually bubbles, but it's super saturated. So the CO2 wants to escape from the lake or reservoir. So it's accumulating under ice. And then as soon as the ice comes off, it's able to release pretty quickly. There may be some other interesting things happening there, but at least in general, as soon as the lake comes off, I mean, as soon as the ice comes off, it can escape. And then it kind of bumps around 100% for the rest of the summer. And that's whether you're above or below is going to depend on the productivity of the lake. But for the most part, the most interesting part of this is that it's accumulating over winter. So the amount, the length of winter, so the snowpack that year will impact how much CO2 accumulates under ice. It could just release throughout the winter. If the ice wasn't there, it would just release. But instead, what you get here is kind of like it's like almost like a burp of CO2 that happens as soon as ice comes off. And then again, kind of like we looked at nutrients before, the same idea here with export. If you combine a concentration by the flow, there's a lot more coming out of the bottom of the reservoirs than the lakes. And then again, what controls it? How long the summer, I mean, how long the winter is, and then how long it's been since ice off to get to the concentration at a given time. And then another factor that's related to whether the lake is turned over or not. Okay, and again, I'm just going to skip this in the interest of time. So basically, I think some of the major takeaways here are the reservoir and lake chemistries and benthic macro invertebrate communities and CO2 concentrations don't really differ between each of the sites that I looked at. Which makes sense because the inputs are the same. The lakes themselves aren't really different. The difference is the release. So it's the amount of water and where it's being released from. And that release does seem to it definitely has an effect on water chemistry downstream. It's at least impacting benthic macrovertebrate communities to some extent. It's not, I think maybe if we had taken more samples, it might have started to separate a little bit, but it's at least related. And then there is an effect downstream on CO2 as well, or the amount of CO2 being released. I think if you were to look at other gases like say methane, you'd probably see something similar. So coming back to relicensing, most of that info went into the pre-application document for the Levining project, which Stillwater is currently working on. It's also relevant to the Bishop Creek relicensing because I looked at South and Sabrina, but those are much further along and actually basically done. But all that research helped inform the key questions because a lot of these projects don't have almost any water quality data, and they definitely don't have good BMI data, good invertebrate data. And one of the questions we actually got in this process was from CDFW was, hey, could you look at benthic macroinvertebrate communities below lakes and reservoirs? And I said, sure, let me point you to a chapter on that. We have that info. So it was great to be able to use that info in this process. So currently, Levining is in this step 2 right now. We're in the middle of studies, which will then be used to inform license conditions. So FERC will impose essentially license conditions on Edison, so to manage it in certain ways if the study results show that it's somehow impacting something that is of concern or protected in some way. Whether or not that's a species, if it's relevant to if water quality is out of State Water Resource Control Board basin plan parameters, they may also impose those requirements. The Forest Service also has the ability to just unilaterally impose conditions. They can sue each other back and forth, but the Forest Service can also impose those. And that's informed by these studies, which is why it's really important to set up good studies in the first place and collect data that's actually going to be useful for creating license conditions like this. So if you want to be part of this, something I do and anybody else can do is sign up to get all of the correspondence between FERC and the licensee. In this case, this is Southern California Edison, and then the agencies. So anything that goes from one to the other in a formal context, you can get. It's public information. You can sign up for this. The link is on the screen here. It's short enough. If you're interested, you write it down. You just make an account and you can add I've listed the projects here if you're interested. And you can sign up for any of the information going back and forth. And then if you see something and you know of information that's not included in one of the documents or something's not being brought up, you can submit a comment. If your comment definitely carries more weight when it comes from an agency, it's if you talk to an agency or some stakeholder that's really directly involved, do that and pass your information off to them. We get a lot of non-substantive comments, which are still useful to get a sense of what people are concerned about. But the substantive comments are really useful because they can, if they point out something specific, it's really helpful for informing the process. So on Levining, the studies that are currently happening right now are water quality, which I'm actually in the middle of. We're working on that this week. There's a stream and reservoir fish population study that's going to happen this summer. There's a creek habitat. So creek habitat is always about stocked fish, essentially, or non-native fish, which is what that focuses on. We also had a request to look for dynamo because that's apparently shown up near Saddlebag. I still have yet to see it. I don't know where it is, but we're going to survey the entire reach of Levining Creek, and we will find it and document it. And then there's another study in the lower reach, so closer to the LADWP diversion, which, if you're going to ask for the projects, for the purposes of the project, the affected area stops at the LADWP version. We can talk about that if you're interested, but that's where the project effects analysis stops on Lee Vining Creek. So upstream of that, they're going to look a little bit at how reservoir operations at Pool Powerhouse have impacted channel morphology. So that's underway. Rush Creek has also just started. This is much earlier, maybe a year and a half behind Lee Vining. So they haven't actually, I think they have draft studies, but they haven't finalized them yet. Bishop is almost done. And then Lundy is starting soon. I want to say this summer, they should be starting. So if you're interested in any of these, definitely sign up to get the information back and forth. Talk to your local NGO that gets involved in this kind of thing, or me, the Stillwater folks, or Edison, and bring up your info and be part of the process. And that's it. And I think I have a little time for questions.
Okay, thanks, Adam. And I'll just remind everyone out there, if you have questions, you can raise your hand, you can type them in the chat, or you can type them in the Q&A box. Any questions for Adam about any of the work that's going on here, or any of the science that he's talked about? Okay, if there's no questions yet, Adam, I'm going to sort of take the opportunity to ask my questions, which
Sure.
are, what you talked about the CO2 and the differences in the CO2 between the lakes and the reservoirs.
Mm-hmm.
Can you go into a little bit more about the impacts that the CO2 has?
Yeah, I don't, John's not here, but if he were here, he would remind me to tell you that it's very, very insignificant. It is a very small component of, and if you're thinking about, say, the carbon cycle, it's a very small amount of CO2. Really, it's I guess, academically interesting. And then it's also interesting if you start thinking about it in the context of, I think it's interesting if you start thinking about it in the context of hydropower as being renewable. If you think about it as, you'd have to maybe do it again in a slightly different way, but look at how does the way the reservoir is managed, does it contribute additional CO2 to the system that would not be released from a lake? And if so, how much of the zero carbon emission that you get from hydropower does that offset? And I think the other thing that would be really important to try to get at that would be also looking at methane, which is, as a lot of us know now, is a much more potent greenhouse gas. And another SNARL student, Elizabeth, looked at that for different reasons, but she looked at methane and found a surprising amount of methane in these reservoirs. And so I think if we were to, or not the reservoirs, in lakes, and I think if we were to look at methane, you would find something similar, and it would be not globally significant, but at least significant in the context of considering these as green energy.
Mm-hmm. And are the reservoirs that are in the Eastern Sierra, how significant are they in terms of providing hydropower, to Southern California?
I'm sorry, I was distracted by the Q&A question. Say that again, Carol.
Yeah, I was just curious how significant the Eastern Sierra reservoirs are for providing Southern California.
So we've talked to the SE operations guys about that, and they're pretty small. They're pretty insignificant in the broader scheme of things. But it seems like where they're most important is, especially nowadays when the grid goes down for various reasons, not goes down, but transmission is limited for various reasons, or PG&E is running PSPS because there's high wind and fire danger, et cetera. So in that parts of the year, if Edison ramps up their power generation, they can supply energy to the entire Eastern Sierra on their own if they need to.
Oh, okay, nice.
So that capacity is there, but on a normal day in the broader scheme of things in the Sierra, it's pretty small.
Okay, Adam, you've got a couple questions in the Q&A box there.
You can take these in order. And hi, Scott. Plankton community composition. I want to say Celia Simons had started to look at that, and she has some data on, I don't know if she followed up on it, but she has some data on plankton communities in some of the reservoirs. But I did not look at that. But I think maybe related to Scott's question, I was looking at chlorophyll A, which is phytoplankton, which I didn't point out, but was related, chlorophyll A export was related to the Benthic Macro invertebrate communities. And like everything else that I talked about, the concentration of chlorophyll A was a little higher at depth. And then combination of that and higher flow at depth meant there was more chlorophyll A exported, which, like Scott is asking, may be providing food for the invertebrate community downstream, especially the filter feeders, the black fly larvae that I was talking about. And David's question, has FERC not reissued any reservoir licenses? No. I'm trying to think of any case where they've ever not issued a license, and I can't think of any. There's some odd cases where the licensee, especially like PG&E, has given up a project, but that's not FERC not issuing the license. That's the licensee having some reason that they don't want it anymore. But about the decommissioning process, that's a slightly separate question. That is Edison saying, or PG&E, whoever. So like PG&E is actually pulling out some dams in some other places like Battle Creek, and Stillwater's working on that too. But that's and Potter Valley, so on the Eel River as well. But that's a different question. Basically, the licensee has to say, we don't want this anymore. We don't want to use this anymore. But they have to keep it in at least the short term to say, we're going to, it's basically the same process where you have to say, we're going to look at the impacts of decommissioning and kind of go through the same process as licensing and make sure you're not going to have harmful impacts by decommissioning. And I think that's maybe part of the reason that a lot of the licensees don't give these up, because it's expensive to do that and kind of hard to do that. Because even if they only generate a little bit of power, it's useful at certain times, and it's kind of hard to decommission. So even if that, whether or not that means actually pulling out the dam, that you just stopped using it, it's kind of hard to do that too. The last part of that question though, how much weight does FERC place on environments versus power and water harvesting? That's a complicated question. And it really depends on who's involved. Like I said, FERC is basically just the mediator. So how much emphasis gets put on the environment versus and water quality versus power generation is basically a question of what agencies get involved. So if, say, Water Resources Control Board isn't present for the discussion and they don't express any concerns, FERC isn't going to be aware that there are any concerns, and they're not going to take that into consideration. If nobody brings it up to them, they don't know to consider it. And it's the same way with the environment. And that's part of the reason that there's always so much emphasis on non-native fish, is that's in a lot of cases, that's the mission of CDFW. And so they, not always, but they focus on that quite a bit. So that's where a lot of the concern is. And that's why there's often, honestly, that's why minimum in-stream flows get set the way they do, because they're set for trout populations, basically. They don't look at BMI populations, they don't look at anything else downstream. It's what flows are enough to support trout, essentially. In some places that, if they're native, that makes perfect sense. But in the Eastern Sierra, it's basically to support trout for fishing, is what they're trying to do. Kevin's question, is water clarity an important measure? I've been going on a rant about water clarity out in the field lately because it's measured in a terrible way. And the Lake Tahoe data that you see is great because there's a long-term data set and they make nice little plots and they show you, it's a great sense of what's happening in the system. And that's really useful for Tahoe because it gives you a sense of what's happening in Tahoe from year to year. And they've been doing it for whatever it is, 40, 50, 60 years now, something like that. If you actually want to understand something more in depth about the system, the way they measure water clarity at Tahoe is not really useful anywhere else. It's kind of generally interesting, but it doesn't really tell you that much. I think basically what it's somewhere like the Sierra where you don't have inputs of mud or dirt or anything like that. Basically what it's telling you is how much phytoplankton is in the water. And kind of what you're doing is eyeballing that. That's not exactly right, but that's kind of what you're doing is how green is the water? Like we saw that today at Tioga. Water was a little green. The depth, the water clarity was not as deep as you might expect somewhere else. And so if you want to know something about productivity, you can measure that. If you want to know something about how far water's light is actually penetrating into the water column, you can measure that directly as well. And we did that. I didn't get into those data at all, but we did that as well by measuring that directly. So it's really useful in Tahoe for context about what's happening in Tahoe. But if you went back in time and started collecting it in Sabrina for the last 60 years, it probably would be useful. Starting it now, not that useful. And Stephanie has a question. Significant release of CO2 near Horseshoe Lake. That is a completely separate issue. That's related to the volcanism at Mammoth Lake, or at Mammoth Mountain. I don't know that we would know exactly if our CO2, I think we would probably see bubbles, or not bubbles, but we'd see sporadic bursts or something of these, like I think you do at Horseshoe Lake. But so the Horseshoe Lake situation is related directly to Mammoth Mountain and the volcanism. And I had one lake that's actually pretty near Mammoth Mountain, Crystal Lake, if you've ever done that hike above Lake George. But I don't think we have any reason to expect that the CO2 in that lake is related to like what's happening at Horseshoe. And certainly not at the other ones, which are from Lee Vining all the way down to south in Sabrina. So I don't think we have any reason to expect that the CO2 that we were measuring was related to the volcanism at Mammoth. And that Horseshoe Lake example is a pretty unique circumstance.
All right, well, thank you, Adam. Thank you for all this information, and I'm glad you're back here in the Eastern Sierra, continuing to do your work. And thanks everyone for tuning in to the seminar series. It's been, sadly another year where we're virtual, and keep your fingers crossed. Next year we'll be back in the Page Center in person. But if you missed any of the seminars and you want to view them, Adam's will be up on the YouTube channel tomorrow, and all the other ones are up there. So you can go back to those and view any that you missed. And thank you so much, Adam. And it was a pleasure hearing your work.
Thanks. I'm glad to have a chance to actually present this to everybody who helped me actually work at SNARL.
Okay. Good night, everyone.